Namaste students. So Vidya, I JVN Dr. Vidyapeeth, Assistant Professor Jayoti Vidyapeeth, Means University, Jaipur. Okay. In today's class, we will be going to discuss from the subject Agriculture, Biotechnology, and today's session will be on use of antisense RNA and other technologies. Okay. So let's begin with antisense RNA. So during mRNA synthesis, DNA strand is transcribed to give message RNA. Okay, what we call as mRNA. Okay, and this is the messenger. Okay, RNA is called as sense RNA, antisense RNA. Okay, this messenger RNA is called as sense RNA. Okay? Antisense RNA is a short single-stranded RNA, okay, which is transcribed by a specific gene within a cell. Okay. So, antisense RNA is a single-stranded RNA that is complementary to a messenger RNA, mRNA, okay, strand transcribed within a cell. Antisense RNA introduced into a cell to inhibit translation of a complementary mRNA by base pairing to its creating barrier to the translation machinery. Okay. For example, Shook system of E. coli R1 plasmid. Okay. So this translation arrest cause reduce the amount of protein expression. Okay. Well-known examples of GM plants produced by this technology are flower sour tomato, two cultivar of the ring spot resistance papaya. Okay. So when we see what is the difference between antisense technology and RNAi, so the intended effect in both will be same. Okay. Example, gene slicing, but the processing is little bit different. Antisense technology degrade RNA by enzyme RNase H, while RNAi employs the enzyme DICER to degrade the mRNA. Okay. So RNAi are twice larger than the antisense oligonucleotide. Okay. So when we see the nature of our nature of this antisense system, there is hook HOK for host killing and SOK for suppressor killing system in R1 plasmid in E. coli. When E. coli cell undergoes division, the daughter cells inherit hook gene and Shook gene from parent, but to short life of the cell, the Shook gene gets degraded. Okay. So in normal cell, hook gene get overexpressed and cell get die. Okay. But when R1 plasmid is get inherited, it having the Shook gene and Shook promoter. Okay. Then it transcripts Shook gene and it get overexpressed against hook gene. Okay. So the mechanism here is in this technique, short segment of the single stranded RNA are introduced. These oligonucleotides are complementary to the mRNA, which physically bind to the mRNA. Okay. So they block the expression of the particular gene. In case of virus, antisense oligonucleotide inhibit viral replication by blocking expression of integral proviral gene. Okay, so usually consists of 15 to 20 nucleotides. Okay, So the translation of mRNA may be blocked by two possible mechanisms. These are by base-specific hybridization, which prevent the access by translation machinery. Okay, that is hybridization arrest. Or second, by forming DNA or RNA duplex, which is recognized by the nucleus. RNAs H specific for digesting RNAs in an RNA and or DNA duplex. Okay, so RNA-H is a non-specific endonuclease, a catalyst that cleavage of the RNA via hydrolytic mechanism. Okay. So when we talk about the characteristic of antisense oligonucleotide, they are unique DNA sequence, efficient cellular uptake, mineral non-specific binding, meaning there is like a minimum non-specific binding take place, target specific hybridization, non-toxic antisense construction is there. So the approaches are, the antisense technology can be modified in three mode because of the chemical modification of the oligonucleotides. These modes are due to the activation of RNA H and internucleotide linkage which do not activate the enzyme. Okay. So first approach, it can be antisense oligonucleotide bind to the target sequence causing both hybridization arrest and RNA H activation. Okay. So degradation of mRNA by RNase H results into the release of oligonucleotides. They may bind to other copies of target mRNA. These oligonucleotides are also susceptible to other nucleases. Okay. This major parameter affecting the catalyst mode of degradation. So second approach is in which the antisense oligonucleotide bind to the target sequence resulting in translation arrest, but they do not activate the enzyme RNase H. Okay. So oligonucleotide and other analogous deoxyribose nucleotide. Okay. So various non-phosphate and phosphate internucleotide linkage falls in this category. Okay. They show resistance against the nuclease enzyme and never get degraded by them. Okay. So they also show effective translation arrest here. A major problem in this, they are generally required higher molar concentration than those of the activate RNase H. Okay. So third approach is in combined feature of both first and second. Okay. They contain both internucleotides linkage, which are responsible for RNase H activation. Okay. Which shows resistance against them. Okay. Then digestion of mRNA targeting DNA, RNA-DNA duplex. Okay, releases oligonucleotide which are resistant against nuclease enzyme. Okay. Hence are more effective than oligonucleotide in the first approach. They may be form hybrids of oligonucleotide and oligonucleotides. Okay, the antiviral activity of an antisense oligonucleotide depend usually on the binding of this target nucleic acid. Okay. So ribozymes are coined by the Thomas and co-worker, and these are RNA molecules which have catalytic activity which degrade nucleotide. So ribozymes bind to the target RNA moiety and inactivate it by cleavage and phosphodiesterase backbone at the specific cutting site. Okay. So the ribozyme destroy RNA that carries the message of disease. These are effective used in HIV virus. Okay. So here the application of antisense like flavor, tomato, antisense RNA used against enzyme. Okay. Okay. And transgenic ACMV resistance cassava plant. Okay. That is African cassava mosaic virus resistance in cassava plant. Okay. Then for so this antisense technology shows a potential for diverse application to field of basic research and therapy. Okay? So it can most approved appropriate in inactivating of specific single specific disease. So antisense RNA can still lack effective designs, biological activity, or effective route of administration. So it can be a powerful weapon for studying the gene functions and for discovering more specific target treatment of disease. Okay. Thank you. This session is powered by digital version 2.0 Jyoti Vidyapeeth Women's University. And hope you are satisfied with my digital session. If you have any queries, please mention in the comment box. I will resolve it. Thank you. Namaste.